Hello, everyone. Good evening. Thank you for being here. I am Manasvi. I'm a PhD student here in NCBS, and I'm standing in for the team at the archives at NCBS today. On behalf of the campus and on behalf of the archives at NCBS in particular, I welcome you all to the 49th edition of the Archives Public Lecture Series. Many of you might be familiar with this forum and the archives at NCBS, but I'll share a couple of lines for those of you who might be new to this. The archives at NCBS is a public collecting center for the history of science in contemporary India. It has over 100,000 processed papers, photographs, equipment, recordings, and much more. If you have time, do check out the space in the basement of this building, the Eastern Lab Complex, just around the corner from this lecture hall. One of us would be happy to give you a tour of the space. You can also check out the website, archives.ncbs.res.in. As part of its programming, the archives runs this public lecture series, framed around discussions by individuals giving talks based on their vast body of work and experience. This is a monthly public engagement to initiate dialogue and debate on an array of diverse topics and histories of ideas. In the past, we have had discussions led by artists, archivists, academics, lawyers, teachers, journalists, and many others. Today, for this 49th edition, we are honored and delighted to welcome Neeraja Sankaran to our campus and to this lecture series. Neeraja is a historian of science writer and editor whose academic life has paralleled her academic interests in the recent or near contemporary history of the life sciences as well as medicine. Within this broad terrain, she has done research and published papers and books on the history of virus research, molecular biology, immunology, and the origins of life research, to name a few. She is the author of A Tale of Two Viruses, which was published by the University of Pittsburgh Press in 2021, last year. This book forms the springboard for this talk. She has also co-authored a history of the development of medical virology in the Netherlands. Her talk today is titled Historical Adventures in Virus Hunting. She also wanted me to mention that she will be teaching 2 courses next semester here in NCBS, one on science writing and the other on the history of science. She will have time for questions and answers after her talk, and the conversation can continue outside over refreshments. Neeraja, thank you very much for being here and for all your work. We are looking forward to hearing your talk. The floor is yours.
Thank you so much, Manasvi, and thank you everybody for inviting me. It's a real honor and pleasure to be here. I'm going to, the modernist poet, certainly one of the greats in the 20th century, T.S. Eliot once said about writers that good writers steal, they don't just borrow. And I'm going to follow his edict because as a historian, I'm automatically a writer. And so should all scientists be. And so I'm going to steal this from a 1957 lecture, the opening lecture by this microbiologist, Andre Loeff, who who opened the lecture with this. From the letter inviting me to give this lecture, I understood that the subject should be general. The dictionary says general means completely or approximately universal, but it fortunately adds within implied limits. I decided that the limits of universality would be the borderline of the kingdom of viruses. So there's a couple of reasons I chose this, because mainly because it couldn't be more similar. I mean, I would have written this in not so elegant language had I not found it already. I too have been asked to give, when Venkat invited me, he asked that the lecture be on a topic that was general. And I decided that my limits of universality, I love that expression and I wish I could use it more, but this is the only time I will, would be the viruses. And so like Loeff, I'm going to be talking largely about the concept of virus. Now, despite the admonition to keep it general, I think given the context of this audience, all of you are researchers, of one sort or the other at an institute for biological research, we all are pretty much on the same ground when we're talking about viruses. All of you know more or less what I mean, if not precisely what I mean when I say the word virus. And you might wonder why there's a whole lecture on the concept of virus. And I'll get into this whole notion of concept versus term as well later in the talk. But I just wanted to start off with saying, although that's true, and most of us, or at least I can remember learning in high school or a little before what a virus was, and we learned a whole lot of characteristics, that they were invisible little things that lived on the threshold of life. I remember that being said. They had either DNA or RNA. They were obligate intracellular parasites. This was a term that we learned back then. Basically, they had to have a living host in order to show signs of their own life. So they lived on the threshold of life, in other words. They could be manifest as inert, not dead exactly, but non-living things. And that they needed a living host to metabolize, live, reproduce, basically show the characteristics of life. All these characteristics were first provided in this particular lecture by Loaf. And this incidentally was a public lecture, as it says here, a memorial lecture at an annual meeting of microbiologists. So this is how he defined a virus, and he may use jargon, he may have used jargon despite his admonition to be general, that may be archaic now, but a lot of it, the basic tenets of how we define a virus are in there. Viruses should be defined as strictly intracellular and potentially pathogenic entities with an infectious phase. And pathogenic and infectious are meaning two different things here. Possessing only one type of nucleic acid, multiplying in the form of their genetic material, unable to grow and undergo binary fission, and devoid of a Lippmann system. Of all the things in their Lippmann system probably needs, may not need too much explanation to this group, except when I was reading it, I'd forgotten what it was. It is something that can produce energy, basically energy molecules or ATP. In other words, again, it refers to its intracellular, its obligate parasitism. All these, so he provided this back in 1957. But this definition appears only midway through his paper. And long before that, he makes this rather famously opaque statement and sort of funny, tongue-in-cheek, where he says, I'm going to defend a paradoxical viewpoint, namely that viruses are viruses. And my ambition is to show that the word virus has a meaning. Before I unpack this, he calls this a paradox, and I wanted to point out the paradox that viruses brought me here, and it also keeps away, it's a virus that keeps the person responsible for me being here, Venkat, out of the room today because of COVID, that particular virus that has dominated our lives in the past 2.5 years. But what did he mean by viruses are viruses? In making this statement, it's sort of like saying a rose is a rose. All of you are familiar with that, or dogs are dogs, or cats are cats. That viruses were a category of being or an entity apart from anything else. That the word virus has a meaning, and he used his lecture to prove that. My ambition is slightly different. My ambition is more like an invitation to come with me back into the past and not just talk about what is a virus. He asks that question before he says viruses are viruses, but to go back, turn back the clock and talk about what was a virus, and to sort of show how the meaning, how the meaning evolved over time to finally arrive at the definition provided in 1957, which is basically how we still define viruses, at least in biology. So one of the things, and I'm drawing on a historian whose work I draw on a lot here, I'm quoting her. You don't need to really read the whole thing. I just put it up there. I'm just going to read out bits and pieces of it that are pertinent to our talk today. She makes the statement that scientists, scientific terms do not possess a single well-defined meaning. Meanings are not constant. And moreover, the scientists who use these words themselves may not perceive these changes in meaning. and that this is not necessarily a bad thing. So often when you're talking about things, you think you're talking about the same thing, but if you probe it, you're actually looking at different things, and parsing out those differences actually leads to making new knowledge. It's all very interesting and theoretical. I just brought it up. The author's name is Ilana Louis. She's a historian and sociologist of science. And the point she makes is different meanings of a concept often coexist in different communities at the same time. And with virus, that's certainly true. I mean, here in a biology institute, we all have a common understanding of what a virus is. But if you go back and look, even in a more general audience, in a more general usage, people have been talking for 30 years of a computer virus, of something that gets into a computer and invisibly causes glitches in the computer. There's also, since the advent of social media, the expression going viral, an expression that itself has gone viral, if I may, using a very reflexive, self-referential sort of analogy. It's gone viral. And both of these definitions or uses of the word encompass some aspects of the biological meaning of the word virus, but not all. The computer virus refers to something that's able to cause glitches or cause disease, which is not even a fundamental property of viruses. They may cause disease. There are lots of viruses that don't. Going viral is this ability to spread very quickly in a community. So the more interesting thing, and what I'll be dealing with when I said come with me on a journey over time, is the meaning of the virus as it's changed over time. So it first was imported into the English language from Latin sometime in the 15th or 16th centuries as a word for poison or venom or noxious substance. And it's used literally, physical poison, those were viruses, but also metaphorically, which I found very interesting, to describe poisonous thoughts and ideas. The first appearance in the scientific text, and you may recognize this picture, was in 1798, or one of the first known in an English language text. We are talking about the use of the word in English. And it's a very, very long treatise title. An inquiry into the causes and effects of variola vaccinate, a disease discovered in some of the western counties of England, particularly Gloucester, even though it's spelled Gloucestershire, and known by the name of the cowpox. The writer was Edward Jenner, and this is quite an iconic picture of him transferring the cowpox. And I put in a little bit of a facsimile of the page from his original paper. For one thing, I thought it was really cool that the writing is so different, because all their S's, or many of their S's, look like F's. But it also tells you the highlighted word, morbid matter of various kinds absorbed into the system produce effects that are similar. What renders the cowpox virus so extremely singular is that the person affected is forever after secure from the infection of smallpox. In his treatise, the word virus has several meanings actually in Jenner's time. So it isn't just a poison, it is the material itself, the morbid matter. And another place, the italic parts are quotes from the paper. 2 small pustules on the wrist arose from the application of the virus. So basically taking the liquid from the pop, that was the virus. It's also referred more generally as the cause of the disease, that the cowpox virus and the variolous, which is the name for the smallpox virus at the time, it's least probable that the same person would resist the action of both. It's not in the least possible, he says. And sometimes the meanings are quite ambiguous. Not only the heels of the horse, but other parts are capable of generating the virus. Generating the fluid, yeah, sure, but also generating the cause of the disease. So it's ambiguous and multi-meaninged at the time, in Jenner's time. Very few people thought at the time to associate any of Jenner's discoveries or his claims with a discovery from 100 years ago made by this man, Anthony van Leeuwenhoek, a haberdasher, a cloth merchant, who of exceptional curiosity, very curious, and subjected everything to examination. And using this paddle-like instrument, which is a microscope, and in here is this tiny little bead that just about a little bigger than there is the actual lens. And he looked at all sorts of fluids, scum of the teeth, water in which he ground up pepper, and wrote a series of letters to the Royal Society describing what he called animal cues, living things or things that he believed were living that were showing motion, and these were microbes. So he is credited with being the father of microbiology, certainly the first person to describe in such detail bacteria or little germs, and nobody connected this with anything that Jenner was saying about disease. For that, one would have to, this was back in 1683, so 100 years before Jenner. In order to make those connections, one would have to wait nearly 100 years after Jenner to culminating in what's called the germ theory of disease, where all germs were considered, many infectious diseases were attributed to specific germs or bacteria. And Louis Pasteur was one of its chief architects, along with Robert Koch and Joseph Lister. And he made the statement in a treatise about rabies, ironically a virus disease, where he says, and forgive me if I mangle the French, but I'll try it anyway. En résumé, tout virus est un microbe. In summary, every virus is a microbe. And what did he mean by that? Microbe is another word for germ or bacteria, particulate. But for virus there, he's using it as a synonym for an disease, causative agent of an infectious disease. A long phrase, he uses the word virus. So simply that all infectious diseases are caused by specific microbes. That's kind of the gist of the statement. This is the sense in which it had become, the word virus was being used, or the concept of virus had begun to be generally used by the late 19th century. So over in the Netherlands, at first, for example, just a few years before he made that famous statement, a scientist called Adolf Meyer, heading an institute, started to investigate a disease of tobacco plants. Now, this is a time when farmers' concerns were very much translated into what was being done at a university. So he was at a research institute, and farmers sent him tobacco plants because their crops were suffering, and he started to investigate the disease. And based on what he was able to find out through how they were infecting stuff, his conclusion was that the Mosaic disease is a bacterial disease, which, however, the infectious forms or viruses are not isolated, nor are their form and mode of life known. So he assumed it's a bacterial disease because of the characteristics. It's being called, it could be transmitted from one infectious plant to an infected plant, excuse me, to a plant that was showing no signs of the disease. It could be transmitted yet again. You could transmit it by filtering the sap and of the infectious material, but nothing was held back. He couldn't find the bacteria like you could in other diseases. His work was taken up a few years later by a graduate student in St. Petersburg, Russia, and he went on to say that the disease was explained most simply by the assumption that the bacteria were secreting a toxin, which is dissolved and therefore it goes and causes the symptoms. So this at first was the idea. Similar ideas were also being put forward in case of animal diseases. Around the same time, a pair of scientists, disciples, workers with Koch, were investigating something called the foot and mouth disease of cattle. Again, a massive economic disaster. They too identified the disease as a bacteria, and they said it's due to the presence of a causal agent capable of reproducing. And the notion of reproduction as being important sign of life, a living cause, is they make that very clear. But they also said explicitly that the agent must be so small that the pores of a filter, which would hold back the smallest known bacterium, would still allow it to pass. So their interpretation is slightly different than that of Ivanovsky, who said there's a toxin. They're saying, no, no, there is the actual bug, but it's going through. It's really small. It's there, we just can't see it. We need a better microscopes. Few years later, another person, unbeknownst to Ivanovsky, in the same institute as Meyer, who had been interested by Meyer's work, but decided I couldn't do anything about it right now. I don't have the time or the right techniques, went back to investigating it. And I apologize, but that's the only, apologize for the blurriness of the quote. I'll talk about it. His name is Martinus Beijerinck. And in 1898, he went back to the tobacco mosaic virus disease. And he said, look, it is a disease, it is an infectious disease, but it's not caused by microbes. Rather, it's caused by contagium vivum fluidum. And I'll break that down for you. Contagium refers to the infectious principle, vivum to the living nature of the disease agent, and fluidum to the fact that it was fluid or dissolved versus particulate, which is what bacteria were. He describes bacteria or microbes as contagium fixum. Very quickly, also, I should say that he used this term contagium vivum fluidum in the title of his paper and in subtitles of different sections. But within the paper, when he's talking about the causative agent, he refers to it as a virus. So the term virus is already being used. But he's using virus as a generic term for all, and the contagium vivum fluidum, he says, is a specific type of virus that is not a bacteria, it's different. And why did he say it was fluidum? Well, he did these experiments on agar. He was able to use the solid medium and extract the material, the infectious material, spread it on the surface of the plate. And his argument was, if it were particulate, the virus, it would remain on the surface. It could not diffuse because the pores of the agar are sizes of molecules. The deep layers would not become virulent, but a water-soluble virus ought to be able to penetrate. And he shows by sterilizing the surface and drawing material, he was able to show that it was in fact going all the way down. So he decided it was fluidum. Why did he say it was vivum? Same there, he's in agreement with Loeffler and Frusch. And he says that because you can extract it from one plant, put it in a new plant, get a disease, extract it from that plant and put it in yet a new plant, there's clear signs of its reproduction. And somebody challenged him when he made this presentation at the meeting, none other than Hugo de Vries, famous and very influential botanist and scientist in the Netherlands at the time. And Beijerinck retorted that he considered the ability to reproduce to be the major characteristic of life. So again, he's agreeing with a lot of things that Loeffler and Frusch said, but disagreeing, and very directly, they actually had some debates about this in writing, that it was bacteria versus a virus. He also made a few other statements, and then I'll move on. And this is a statement, I wish it were clearer, because this is something that as an historian gave me goosebumps, literally, because in here, he's bringing forth an idea which was so outlandish at that time that people didn't know what to make of it, and they just put it aside for the next several decades. And what he's doing is saying that virus is the reason why a TMV, the contagium vivum fluidum, that the virus of tobacco mosaic disease is different from a bacteria is because of, and he's describing it as obligate intercellular parasitism, or the threshold of life. He says it's only living because only organs of plants that are growing and whose cells are dividing are capable of being infected. And only in these dividing cells does the virus reproduce itself. And that is something that was, like I said, so bizarre that now it's something we take for granted, that viruses only divide in living cells and typically use active the machinery of their host cell, right? He also goes on to say the virus may be dried without loss in strength of infection. The virus may winter in its dry state. What he means by that is that it's inert outside of a living cell. So it winters and it does not show any signs of life. It's hibernating, so to speak. But you put it back in a living cell and it will be as infectious as it was before. So there's Byring. People didn't know what to make of him. And also he moved on to other things because, you know, the Dutch industry, the tobacco industry literally died by then because the disease was causing enough trouble and they moved on and tulips became big. And tulips, even when infected with viruses, looked pretty and were ornamental plants. So you didn't really, it didn't matter that they didn't produce enough things. So, you know, and the Dutch are still famous for tulips. So, and he became famous as a soil microbiologist, so he moved on. But I'm going to move on to two other discoveries now. But before doing that, just say that one common thing between all these scientists was that the agent was filterable, whether the agent itself or the disease principle, that is a toxin, something there was filterable. And by the early 20th century, the term that set aside all these entities from other infectious entities were not just virus, but very specifically a filterable virus, that it could not be held back by filters and it was passing through. So it was really, really tiny. And now I'll go on to the next important discovery in 1911. Peyton Rouse, that's the scientist. He is a new, he was in 1911, 1910, he had a new employee hired at this institute, the Rockefeller Institute, which some people here in the audience, or at least at the institute, would be very familiar with. I worked there myself as a writer and historian, but our director here got his PhD there. But back in 1910, 1911, Peyton Rouse had just been hired at this institute, which was explicitly not a cancer research institute, but Rouse had been hired specifically to do cancer research by the director, Simon Flexner. And a worried farmer brought in this hen. Well, not exactly this hen, but a hen that looked something like this with the globular mass protruding from its breast. I can't imagine that if a worried farmer came through the gates of NCBS with a cow that was in great pain, or a donkey or a goat would be let in. This is in the middle of New York City, and he was let in 1910. So that itself for me is great. But he was brought and Rouse started his investigations. And he found out very quickly that this tumor was caused by something unusual, unknown. It was, you know, not caused by any previously, was not like any previously known tumor. Because not only could you transplant it to new birds, provided the birds were of the right species, by putting bits of the tumor, but then you could do pretty much what Byring and Loeffler and all the others before him had done. You could grind up the tissue, filter it, and transmit it with the filtered extract. So his conclusion ultimately, within the next year or two, was he described it as a minute parasitic organism. In other words, a virus. He very deliberately was prevented from using the actual term virus or even filterable virus by one of his senior people at the institute for a number of reasons. Cancer was not believed to be an infectious disease. And the stigma, literally, of associating it with a virus implied whether or not it was true that it was and that it was an infectious disease. And it had just, not only was it not believed, just the previous year an international congress had said that any parasitic theory of cancer is simply not true. It had arrived at this consensus, and his senior, who was less brash than him, said, look, if you want to publish your results, if you use the word virus, it's probably not going to be accepted for publication. So he says it's possibly a minute parasitic organism. The other thing that happened is that one of his very close colleagues was not exactly, who joined him within a few months, was not exactly in favor of the idea of a virus, but I'll get into that in a few minutes. So there was one discovery. A few years later, across the pond, at this institute, the Pasteur Institute, that's not how it looks anymore. I mean, that building is probably still there, but it's flanked by other buildings and whatnot. This is how it looked back in the day. This scientist, a French Canadian, himself having crossed the pond, or at least a French-born, Quebec-raised self-taught scientist, very interesting man, Felix Derel. He had been charged with investigating an outbreak of bacterial dysentery in a garrison. And through the course of investigating that, stumbled on this phenomenon where he found that bacteria that were causing the disease were being lysed by something that he thought was a virus, and that it was a transmissible lysis. So you mixed it with that substance that he was able to get from the intestine of patients. Long story for which I don't have time, fascinating story actually. And he described it in French, which is why the words are flipped, and we might have used it in English, as the cause as a microbe invisible microbe that gave a name to bacteriophage. He wasn't the first person to describe this phenomenon. 1915, somebody else already had described it, but not in dysentery bacilli, in a different bacterium. But he's the guy who gave it its name, bacteriophage, and for the rest of his life worked on the bacteriophage. A contrast with Rouse, I should say, who, having discovered the filterable, the sarcoma agent, stopped working on that problem in 1915 for a variety of reasons and never went back to it. So these two things were and there's some remarkable parallels, actually. They were both transmissible phenomenon, transmissible biofilterable agent. They both were conceived of as filterable viruses by their discoverers. The reception was a fairly uniform, or largely, they had a few supporters each, but on the whole, people said no. And the disagreements, other than cancer cannot be an infection, hinged on very similar issues of, those are the parallels actually so here are some of the oppositions. This is James Murphy, who was Peyton Rouse's really close colleague, having joined his lab in 1912, and really is the person who carried on sarcoma virus long after Peyton Rouse had left it. But he, in a letter to a former student, says, I never believed in the virus theory. And in writing about it a few years later, he said, the hypothesis that the specific principles of chicken tumors are comparatively simple substances which belong to biochemistry rather than to bacteriology, which is a fancy way of saying that the agent is not a virus, it's coming from within the cell. And he said later in the same paper, the ability of such agents to induce permanent inherited changes may justify differentiating them from viruses. So he is completely against the idea that cancer sarcomas could be caused by viruses. A very influential opponent to Derel's work was this man, Jules Bordet, who in 1922 already wrote this. According to Derel, lysis is due to a living being, a filtering virus. We, on the contrary, believe that the lytic principle originates from the bacteria themselves, which, when touched by the substance, are capable of regenerating it. And he persisted in this idea for much longer, and as late as 1931 said, the invisible virus of Derel does not exist. Very similar lines of argument of the two men. Bordet would play a bigger part, and I'll come to that very quickly. So I was struck by these parallels. I first noticed them when I was working on my PhD, when I was actually working in this gray area that's not really detailed in this picture. But I was struck when I went back to say, hey, I was studying bacteriophages at that time. And I had already studied the Rouse story as a science writer, proto-historian at Rockefeller. And it struck me that these are very similar. And in fact, the stories did go on to be very similar. The reason for the solid line represents the kind of research being done on bacteriophages. They were a brand new discovery in 1917, and this kept going actually. The reason for this dashed line is to show that there was a kind of stuttered progress of cancer research. Cancer had been known for much longer, of course, and would continue, and it did not really resolve until the late 70s. But the sort of parallels struck me that conceptually, at least, the way these things played out were very parallel, and I started to investigate that a bit more. And the converging point for the parallels, we have to go back to Loaf, who in 1953 wrote this fantastic review based on work that he'd done. Those papers were written in French, this was written in English, about a phenomenon called lysogeny. I'll try to explain what lysogeny is, but I may not have time. But let me go on to say lysogeny is very paradoxical. It played a very paradoxical role in virus research because it went at first as the biggest hurdle, from being the biggest hurdle in the 1920s to accepting the notion that bacteriophages could be viruses, to becoming a linchpin that then allows you to bring many entities hitherto undefined into the broader category of viruses. The person who discovered lysogeny was none other than Bourdet. And along with a Romanian colleague, Ciucă, his idea was that lysogeny was basically the spontaneous appearance rather than the continual appearance of this lysis in bacteria. And he said that it's simply impossible that something carries an infection generation after generation and doesn't show any signs of that infection. And then suddenly, boom, one day it does. So he said that it contradicted the idea of an infection of the bacteria by a virus. And he thought, therefore, he continued to say that it's something within the bacteria that adjusts the metabolism. And that's what's causing this lysogenic, this transmissible lysis. Sorry. This resulted in a decades-long impasse between this notion of lysogeny as an intrinsic factor, contradicting the idea that a bacteriophage could be an external virus. There were people, and this is where I began my PhD research, who suggested, well, maybe there's a kind of phase shift happening and something is switching forms and becoming a virus as well as this intrinsic factor or a gene. But again, this was in the 30s before anything could be done about it, and those ideas, not much notice was taken about them. Meanwhile, elsewhere in virology, a whole bunch of things happen. Electron microscopes, X-ray crystallography, all these techniques and instruments were invented. People began to be able to culture viruses, animal viruses in vitro, or at least in plates. Lots more could happen in virology. You figured out how to play with viruses in the lab, different scientists. And so they could start testing various ideas, both about cancer viruses, sarcoma in particular, as well as about bacteriophages. Although, yeah, you could see the bacteriophages under the electron microscope, for example, and see particles. So all that was happening. And then there was a more general biology advance that was very important. Not just this story. My thesis is the most important discovery for 20th century biology was this. I've given the title of the paper 1944, but it sums it up in a book written by one of its authors some 30 to 40 years later, discovering that genes are made of DNA. So people started to figure out what DNA was. And so these advances made it possible for Loaf to actually prove that lysogeny was being caused by the bacteriophages were existing in this dual mode as viruses or as genes, which people before him, like McFarlane Burnett from Australia, had suggested. But he had suggested it in the 30s, before the 1944 discovery. Loaf was able to show what he said was happening was, here's a bacterial cell, here's a bacteriophage. It has DNA, bacteria has DNA. What's happening in lysogeny is that it's knitting itself, becoming part of the bacterial DNA, so that now it just reproduces. It's become lysogenic. He called this phase of the bacteriophage a prophage, a proto-bacteriophage, so scrunches down to prophage, and he got the Nobel Prize for this particular, or he was one of the recipients of the Nobel Prize. His part was given for this thought. And very quickly, his concept of virus depended a lot on describing lysogeny. And so within 4 years, he'd already given this public lecture on the concept of virus. He brought lysogeny to a head in 1953. But what about the chicken sarcoma virus? That's all very well and good, but it refers to the bacteriophages specifically. Already in this paper, he suggested parallels between the cancer situation and phage lysogeny. And what he says, the neoplastic potentiality of a cell could be visualized or imagined as perpetuated in the form of the genetic material of the neoplastic particle. So basically, fancy language for saying you can think of the cancer causing agent as its genetic material becoming part of its host cell. And if you think of it that way, it's very similar to what's happening. He's only suggesting this, and then he also adds the development of phage in a lysogenic bacterium is lethal. So once the phage DNA is the prophage is stimulated to create a virus, the virus comes out and lots of them are formed and they burst out of the bacteria and kill the cell. The neoplastic agent is not, he used the wrong word, pathogenic for the neoplastic cell. It's basically not killing the cell. Instead, it's causing it to multiply a lot more. It's difficult to see. At the time, you didn't have the right techniques. And so this idea of the chicken sarcoma virus, whether or not it could be lysogenic, had to be held up because culturing techniques for tumor viruses advanced much more slowly. Bacteriophages, in fact, were discovered because they were found in growing bacteria. So the reverse, there's a bit of reversal, so to speak. Also, RNA was an RSV, viral sarcoma virus that it came to be known, was an RNA virus. And this is important because, as I said, viruses were determined and recognized to have only one type of nucleic acid. And by this time, not only was it found that DNA is the carrier of genes, except for the RNA viruses, but also in place quite firmly was this notion of whatever familiar to most people as the central dogma. That is, DNA could make RNA, could make proteins, and I'm mangling this. So there's a whole lecture on describing the, but this is very shorthand for what was central dogma and how it was perceived in those days. And that was a big holdback. In fact, a very prominent player in this whole story was a scientist called Renato Dulbecco, who deliberately chose not to work on RSV, even though he had moved from bacteriophages to cancer viruses. And he waited for a DNA cancer virus in order to show that lysogeny was happening in a DNA cancer virus. But with RNA virus, one had to wait for this guy, graduate student of the aforementioned Dulbecco. His name is Howard Temin. This is him at the University of Wisconsin. As a graduate student in Dulbecco's lab, he, together with a postdoc, Harry Rubin, developed an in vitro assay for tumor viruses. In this, he was following the footsteps of his PhD advisor, who had developed an in vitro assay for animal viruses that could cause harm, so lethal, the same way bacteriophages were killing bacteria. In 1960, already as a PhD student, in his thesis defense, he suggested what he called the DNA provirus, that the RNA genome of the RSV was producing a DNA provirus, sort of like a prophage, that integrated into the cancer cell. But that was where the holdup of the central dogma was. So, and RSV therefore underwent some sort of lysogeny. Took him him 10 more years, a job. He got his PhD anyway because he didn't put that in his thesis. He merely tried to bring it up in his defense. He discovered an enzyme at the same time as somebody else called David Baltimore also discovered that same enzyme. RNA-dependent DNA polymerase, better known as reverse transcriptase, that indeed is able to look at RNA and make DNA. So it was an enzyme that was able to make DNA molecules from RNA templates rather than DNA templates. And that sort of brought the story to an end, to its denouement. So from discoveries, the 1911, 1917 discoveries, to, that wasn't supposed to be 1911, I'm sorry, that was supposed to be 1953. 1953 to the 1970s final denouement. This whole story is what I tell in my book, A Tale of Two Viruses, whose subtitle, very long and completely blurred in the picture, is Parallels in Research Trajectories of Tumor and Bacterial Viruses. I'm not going to talk much more about my book. There is a Dickensian, Tale of Two Viruses is clearly borrowed from Dickens. I've also used other Dickens tropes in my book, but the one that I wasn't looking for, and if I have time, I would like to mention it. I'm down to my last 3 slides. Is this one, because it's so fantastic. A question of life imitating art is here. Anybody who's read A Tale of Two Cities, and if you haven't, you really should. It's a great adventure story set in the French Revolution. Will remember a famous villainess. To me, one of my favorite literary villainesses. This very chilling woman who sits at every guillotine. Whenever prisoners are being led to the guillotine, she goes and sits there, and she's constantly knitting. And what is she knitting? Is a roster of all the victims of the guillotine. The name is slightly rhyming, bacteriophage, but in lysogeny, that's pretty much what's happening. Bacteriophage is becoming a Madame Defarge, because what it's doing, and a very talented microbiologist, cartoonist friend drew this for me, is that the bacteriophage is taking its DNA, knitting it into the DNA of the bacterial host, and thereby earmarking the bacteria as a victim of the guillotine of lysogeny, bacterial lysis. And so my friend drew this cartoon with a background picture of the actual guillotine, cartoon of the guillotine. So I was really thrilled with this, so I thought I'd throw it in, even though it didn't have, and it was an adventure with viruses. I'd like to finish my talk with a nod to the particular context in which I'm giving it. I was invited here by an archive to give a public lecture. And so where do archives fit into this story of the historical adventures of viruses? And I'm going to say absolutely central. I'm not going to leave it, I'll just explain. These are various pictures of the archives I've visited physically. Again, not necessarily in the current form in which their pictures are shown. This here is the Rockefeller Archive, a fabulous mansion outside in upstate New York, up the river from New York City, which interestingly does not house the papers of any of the Rockefeller scientists. For that, I had to go to Philadelphia, to this building, across from one of the most famous monuments in the US, the Liberty Bell. This library was set up by Benjamin Franklin, and it hosts this beautiful reading room, which I took a picture of just to show, or I didn't take a picture, I lifted the picture from their website, which is where I sat and did a lot of my research and learned about Rouse, learned about Flexner, but especially about Rouse. They have boxes and boxes and boxes, just like this archive has of Professor Siddiqui, the founder of this institute, for example. And that's where I found some fantastic material that had never been written about before. That was the Pasteur Institute, where I got a lot of information about Darrell, including an unpublished book of his that an autobiography that, as one person says, reads more like a novel, and you shouldn't really take everything completely seriously. So that's a potential future project. This is the Wellcome Institute, again, with its reading rooms. In London, another place where I did a lot of research, procured photographs, finished up my book over there actually. And this is a new library at the University of Wisconsin. And this is a rock named for Temin, on his bicycle. It's actually on a bike path. And they've named the bike path for Temin because he used to ride up and down there. And his papers were housed there. And that archive only came into being in 2016. Until then, nobody had archived Temin's papers. And so if I'd wanted to write, I mean, I couldn't have finished my book before I did, because until 2017, I didn't even get to go and visit those archives. You know, that's when the papers were finally being made. So archives are, and there's a physical joy, there's a joy to going and physically, I mean, those of you sitting in the front row, I'm preaching to the choir, but to everybody, there is something about going to an archive and looking at the physicality. I mean, I will have to give a nod to digital archives. They're very useful. They're very, very essential for people like me who I was able to go to a lot of these places, but a lot more work I did simply by getting sources offline. But there's something about seeing things in their context next to papers that gives you a sense of the story that simply looking at a reprint will not. So archives are absolutely central, and which is why I really want to thank the archive for having me give this talk about viruses. And really, that's it. Thank you.
Thank you.
How do I turn the mic off?
No, we have time for questions and answers.
Oh, okay, sorry.
Please raise your hand and if you have, we'll get a mic.
I'm sorry, a lot of time I was looking at the slides rather than at you guys. I do apologize for that. I don't normally do.
Till the thing, I do have a question.
Please.
What's the significance of the rock?
The rock is just a plaque. It's on the path that Kevin used to bike to his lab and in the University of Wisconsin. And I used to walk up and down it. And I took the picture one day because I was going to visit the Temin archives, and there was a plaque dedicated to Temin. They called it the Howard Temin Way. He died young of cancer, quite ironically, because he resolved how viruses cause cancer. And actually, at his Nobel Prize, he got a Nobel Prize, and he actually admonished somebody for smoking, and then proceeded to die of lung cancer himself, which was not the consistent.
Anyone online? You can.
Yeah, no, there's somebody right there.
Hi, Neeraja. Thanks for the talk. I was just wondering about your story there, how you got interested in studying viruses, particularly, and looking at these people. A little bit more about that would be great.
So it's a story that has several parts. I was making a transition from being a out-of-work writer to possibly becoming a historian, trying to apply to graduate school. But I got a job at Rockefeller. Actually, I'd gone to Rockefeller to apply for a job as a writer. And a guy there asked me, if you didn't have to worry about money, what would you do? And I don't know why I said I want to do a PhD in history of science. And he said, okay, forget about this. I'll give you another one. It'll be a short-term one, but I'll get you into grad school. And as a result of that, I was in Rockefeller working on different projects to celebrate their centennial. This was in 1998 that I joined there. 2000 was their centennial. And that's when I encountered the Rouse story. But it went to a back burner once I got into graduate school. In graduate school, I picked up on a dissertation topic. You have to find a dissertation topic sometime in your first 3 years. I didn't for the first 2.5 years. But at a coffee session with a visiting speaker, she said, Joshua Lederberg, very famous scientist, says that McFarland Burnett did amazing work with bacteriophages that nobody knows anything about, nobody's written anything about. Yes, and bacteriophages were certainly something one knew about Derrell in the early controversy, and one knew about Delbruck and the American phage group, which is in the 30s, but those intervening years, nothing was known. So I started working on Burnett as a thesis topic because it was very confined. There were exact number of years that he worked on bacteriophage. When doing that, I mean, it was a topic that interested me. It seemed like an interesting one, and it was a thesisable topic. very important when you're a graduate student. I think you need to be able to find a topic and a research project, be it in history, be it in science, that you can finish within a finite time, publish on, and get ahead, because your life does not end with your PhD. And at that time, I discovered, and I noticed this parallel, and I thought, hey, cool idea. Then I got a job and another job and a postdoc. I did things in reverse. At some point, I sat down and thought about it. It wasn't in 2007. I got my PhD in 2006. 2011, I said, I better do something about these parallels. And 2011 was also the 100th anniversary of Rouse's. And so we put together, me and other people from other universities, put together a panel on 100 years of cancer viruses. And I projected this idea that there are parallels in the bacteriophage story. A few years later, that became a book proposal, and then eventually. Very long answer, I'm sorry. Can I unmute as he wishes to ask directly? Sure.
Yeah, you can unmute and ask the questions.
Hello.
Hello. Yeah, it was an excellent talk.
Thank
Very nice, very motivational, especially for young people, but I don't find many people out here. I'm from Delhi University and I'm a virologist. Many students, when I teach the virology history part, they ask me, sir, is there any provision of doing a PhD in history of virology? So I think you are the best person to answer that. How many universities and where do they have this program on history of virology or history of microbiology as such? Are you aware of?
It's history of science. I did my PhD at Yale
Okay.
at a time when Professor Larry Holmes,
Oh, yeah.
he was my advisor to begin with, but then he got ill. And then I got Professor Bill Summers, who's primarily a molecular biologist. But you don't go in with the history of virology. You pick your project. You know, unlike a science lab, you don't have to be doing what your advisor did or does. All you need to do is to go in and yak with your advisor about what you're finding out and write something plausible, and you go back and forth on the ideas. They test you on your historiography, but really how you do this is to apply for a history of science program more generally. And you have if you have an idea, this is what I want to investigate, brilliant. I have a ready-made project for anybody who wants to. I mean, I have a few ready-made projects, but I do think a large unwritten part of history of viruses is the history, for example, of virus cultivation and tissue culture, because there's books written on tissue culture as a technique, but how it's harnessed to grow viruses is in fact, it's there in my as in my book. In the final afterword, I've written, are there any graduate students reading this? Please go out and do this. You know, so I think you have to find your projects yourself, but there are a lot of very good history of science programs in the US, in the world. We're hoping there will be some in India. I don't really know of any departments dedicated to history of science over here. I think under a history of medicine, you could conceivably take it in that direction.
Right. Okay. Thank you so much. It was highly inspirational.
Thank you.
Enjoyed it thoroughly.
Thank you.
Any other questions?
Is anybody tempted to shift over from science to history? Do I have any takers?
People here are already quite.
I'm just, yes.
Biology, I come from public health, as you know, and I'm fascinated by the fact that history of the actually support understanding of the movement of science. And in my field, which is public health, it was sort of almost, I think by the time I'd say 2015, 2016, we thought we had come to the end of communicable diseases. as a sort of field that needed to be studied. And you would go on to something called lifestyle or non-communicable diseases. Because it is sort of the sense what we could control these things. What we couldn't control or what we needed to work on was people's behaviors. And there was a huge emphasis and a shift in the way public health moved. And then suddenly we were hit by one after the other of getting back to understanding. And as you said, it's fascinating sort of almost conflict between people who have chosen rather remain within the old hygiene side of public health. As you know, many of our institutions, in fact, the one I studied in, actually removed the word hygiene from its very, Johns Hopkins School of Public Health, which used to be called the School of Hygiene and Public Health. I didn't think hygiene mattered anymore, to do all kinds of antiseptics and things to remove it. So I was just wondering if there are these sort of confluences and changes that you are noticing in your field that have come about because of this. And I mean, from the question that I heard, obviously the popularity of viruses becoming viral in a way has to do with what's happening.
That's got to do with COVID, of course. But I think there's never been one or the other. And I think every time any field sort of says this is the end of something, it is to their own peril and they're proven wrong every time. I mean, we talk about this, forgetting that while people are saying this in public health, AIDS is still a pandemic. Started in the 80s. It's still very much a pandemic. It's a communicable disease. Granted, it's sexually communicable. So there's one very specific way it's communicable. But it's not just about behavior, right? It's also about understanding the virus. And I think it's really, I mean, we have this argument within any discipline about who's the winner and one side taking over the other. And I always feel that if there's a clear winner, the field on the whole is a loser. Because if there's a clear winner of behavior over viruses, you're not going to learn enough about the viruses. But there's no denying that behaviors are so important. I mean, COVID has taught us that. The fact that many of you are sitting here with masks has taught us that masks, it's a behavior, right? People who don't wear masks do so at their, not just imperil themselves, but other people. This is not meant to be a platform for preaching. For masks, I'm standing here without one. But what I mean is, you cannot separate the importance of either or. There's never been an either or. It always has to be both. And it works better when they work together and without these artificial, what I consider largely artificial boundaries between internalists, externalists, which is a big one in my field, but hygienists versus the hard scientists. I mean, you need to work it all together, and that's the only way we're going to solve these problems. But by the way, viruses aren't going anywhere, and I don't think diseases are going anywhere because viruses have been on the planet for eons, literally eons, longer than humans ever have, and they will continue to be there. We need to learn how to deal with them. And if any of you have seen this movie, King's Men, that is not the way to deal with them. It's a very funny movie with people dying with bursting off. It's one of these crazy films. Anyway, I like to bring in pop culture invariably, so I just made that allusion. But it's got to do with trends. Trends are reflective of how people are thinking. But then it's up to the historian to actually take the step back. Historians, anthropologists, sociologists who sort of take a step back from the trees and go to see the forests and say, look, this is what needs to be done. We need to prepare ourselves for the fact that diseases aren't going to go away, especially as people are living longer and longer. Other kinds of diseases will pop up, but that's a whole other body of work. Thank you for your question.
Thank you, Neeraja, again, for that fascinating talk.